Thank you, organizers, for inviting us, and thank you, Aging Connectome collaborators, for describing our study. I'm going to show you how and why we are adding 7 Tesla magnetic resonance spectroscopy to our data set. Over the past several years, while our NIH U01 was being completed, I added the study biochemistry underlying the connectome. In this separate R01, I acquired 7 Tesla MRS from participants who were willing to come back after completing the connectome protocol. Going forward into the U19, we will acquire 7 Tesla MRS from participants within the same grant. This talk will introduce MR spectroscopy, or MRS. Summarize key data from 7 Tesla MRS in aging and AD. These published data were used to train classifiers that were then applied to participants in the HCA. Show how MRS-based classifiers of brain aging and AD correlate with cognition and genetic risk that were quantified from data in the completed U01. Summarize recent work relating myo-inositol signals to inflammation, and show how our state of readiness to add MRS to the AABC. Let's start with an orientation to ultra-short echo time, ultra-high field spectroscopy. On the top left is a spectrum from a human participant. You can see the large NAA, creatine, and choline resonances that are commonly detected at lower field. We also resolve some weakly represented overlapped signals using basis spectra and LC model fitting. Here I show 5 of the basis spectra that go into the model. We created a basis set of pure chemical spectra that includes all of the resonances that we expect to be present. You can see from absence of signal in the fit residual that this fitting process was successful. An important feature to understand about these spectra and fitting process is presence of very small, highly overlapped resonances in the in vivo spectrum. Based on my prior work, I wanted to make sure that we would be able to pick up the antioxidant signal from ascorbate and glutathione. That's why I went to ultra-high field. The increased spectral dispersion helps to resolve and fit the overlapping resonances. It is also important to detect at ultra-short echo time to avoid confounding of our data by condition-specific T2 relaxation. In our published paper on AD, we fit 14 neurochemicals at 7 Tesla. At lower field, fitting 5 is typical. In this publication, we measured the expected AD-associated differences in myo-inositol and total choline. However, we also measured a difference in ascorbate. I won't blame you for being skeptical about accuracy and precision for quantifying small changes in such tiny signals. I was skeptical too. That's why I'll show you a few of the published validation studies. Here are 2 studies that validate accurate quantification of tiny signal changes in 7 Tesla MRS from the perspective of glutathione. For study 1, Minnesota pharmacy colleagues wanted to know whether N-acetylcysteine, a precursor for cysteine, which is rate limiting for glutathione production, would be able to increase brain glutathione concentration. N-acetylcysteine was administered intravenously at a dose of 150 milligrams per kilogram over 1 hour. Brain glutathione was measured prior to NAC. The person came out of the magnet for the first half hour of infusion, then they went back in the scanner for approximately 1 hour. Pharmacokinetic estimates predicted that glutathione would peak during the 1 to 2 hour time frame. Thus, this is when the participant was placed in the magnet for continuous monitoring of glutathione. The result was that brain glutathione concentration increased in all 9 participants, and by the overall average of 43%. This makes sense given that the blood redox ratio peaked at approximately 1 hour post N-acetylcysteine. This study demonstrates that changes in the tiny signal from glutathione can be quantified from the highly overlapped spectra. That is, in which case the change is not visible in the spectra. I know these spectra look the same, and that is the point. The fitting is able to see what the eye does not. For study 2, we started with a spectrum measured from a human participant. Known amounts of glutathione signal were added to and subtracted from human brain in vivo spectra. We studied 10 participants, 5 of whom were retested. The graph shows that spectral deconvolution software was able to pick up a change in glutathione signal very accurately. And you can also see that even when the change in glutathione was 100% subtracted, all of the glutathione signal was gone. Once again, the signal changes are barely perceptible in the spectra, but the fitting is able to pick up what the eye cannot see. To set the stage, I need to tell you about my published findings from 7 Tesla MRS of aging in Alzheimer's disease. We applied 7 Tesla MRS to measure the 14 neurochemical profile in the human cortex in aging in Alzheimer's disease. We studied the posterior cingulate cortex brain region because in AD, the PCC has plaques and tangles, reduced glucose metabolism, decreased perfusion, and weaker default mode network connectivity. Spectral quality was consistently high in all cohorts. Aging was studied via young and cognitively healthy older adults. We studied mild to moderate Alzheimer's disease based on MoCA and activities of daily living scores, and possible AD based on NINCDS ADR day criteria and consensus diagnosis by a team that included a neurologist and a neuropsychologist. Healthy controls were also seen by the neurologist and the neuropsychologist and needed to have a MoCA score above 23. All studies were gender matched and retest studies were included. We found several age-associated differences in brain chemical concentrations that passed our strict Bonferroni-corrected p-value of 0.05 over 28. These neurochemicals are thought to be linked to energy and glial status, neuronal health, and membrane turnover. We also found AD-associated differences, 2 of which are in good agreement with the literature, but ascorbate had not been reported when we began. In 2018, another study that used edited spectroscopy at 3 Tesla also found higher ascorbate in AD. Higher ascorbate in AD seems counterintuitive since we expect oxidative stress to deplete antioxidants. There are many possible explanations, though I am most convinced by a possible link to inflammation. Random forest analysis of the AD versus control data from the 14 neurochemical profile from 2 brain regions, including the occipital cortex, was able to distinguish patients from controls with same sample sensitivity 88% and specificity 97%. The main predictors of AD status were PCC, ascorbate, and myo-inositol signals, both of which may be linked to inflammation. Let's move on to talk about new unpublished data that were collected by adding 7 Tesla MRS to the HCA. MyR01 benefit greatly from the recruitment efforts and MRI data that were acquired in the U01. The MRS sub-study has a focus on AD, and thus screens more thoroughly for absence of incipient AD. Participants must pass inclusion by our study neuropsychologist and neurologist as having no sign of incipient AD. As a mechanistic link, we computed an average resting state heat map from lifespan connectome data and set our MRS VI in 2 key regions of the default mode network, which is known to have weaker connectivity in AD. In this talk, I'll focus on the PCC data. These are some of the preliminary results from adding 7T MRS to the HCA. The MRS study pre-selected 5 focused neurochemicals. The result from 46 participants who were in both studies are plotted on the left. Some of the neurochemicals can be seen at 3 Tesla, whereas others can only be seen with special techniques such as 7 Tesla. There is an apparent age course for most of the neurochemicals with a noteworthy transition around age 50. These data are in agreement with our published binary age group study. Since the R01 had an AD focus, it collected amyloid PET from participants age 60 plus. Remember, these participants are cognitively healthy according to our neurology team. It is well known that apparently a third of cognitively healthy older adults are PET positive, and approximately a third of participants diagnosed with probable AD are amyloid PET negative. Therefore, it is not surprising that a third of the cognitively healthy older adults in this study are amyloid PET positive, as read by our neurologist co-investigator. Though we only have N equals 16 participants, this finding from PET is the strongest in our study so far. The finding is that NAA concentration, generally considered to be a marker of neuronal health, is higher in cognitively healthy participants who are amyloid PET positive. It is possible that strong neuronal health in these participants is protecting them from the detrimental impact of amyloid burden. This animated slide starts with the outcomes of unsupervised principal component analysis, or PCA, from the published aging and AD cohorts. After I explain this first step, I will overlay data from the HCA. As I described previously, we scanned 16 young and 13 cognitively healthy older adults using 7 Tesla MRS. These are the outcomes of unsupervised principal component analysis. Open symbols represent cognitively healthy individuals with squares demarcating young and circles older adults. Though we took great care to hide all hints on age from the model, the young and older cohorts separated perfectly on the first principal component. We also ran unsupervised PCA on the participants with Alzheimer's disease and healthy controls. Here, circles still represent older adults, though filled red symbols demarcate participants with AD. The separation was good but not perfect, with the first 4 principal components correctly separating AD from control in 67% of the cohort. Recall in the random forest analysis, which used supervision, the same sample sensitivity and specificity were 88 and 97% respectively. The first thing we did was to see how the participants with AD compared to the healthy aging cohort. The method we used was to calculate aging principal components 1 and 2 using the formula that was generated by the PCA of the published healthy aging data. The red circles for the participants with AD are now shown on the aging graph. While some participants fell within the normal range, it appears as if AD is often beyond the extreme of aging, or perhaps AD has its own domain. We similarly calculated the AD principal components from the young adult cohort. These young participants fell within a separate domain from AD beyond the edge of aging or AD in the expected direction. Now let's add data from the Connectome cohort in the aging case on the left. Once again, we use the principal component formulas from the published cognitively healthy aging cohort. As you might expect, much of the HCA data fell in the gap between the age groups. Also, as can be expected given that we only included cognitively healthy adults in this sub-study, none of the HCA participants showed brain chemistry that were in the domain of AD. There's at least one HCA participant who had what looked like 20-year-old brain biochemistry, even though they could not be younger than age 35. We similarly plotted the HCA data in the AD-derived case. Once again, none of the HCA participants fell in the domain of AD, and a few of the HC participants evidenced biochemistry in the young adult domain. To further show HCA data in this approach, I will carry a copy of these graphs into the next slide. Before I advance, notice that a more negative or leftward principal component 1 is in the direction of older brain age or in the direction of higher AD propensity. For this analysis, I consider principal components derived from brain chemistry, i.e. MRS, to represent a composite metric of brain health. During the writing of the AABC, our collaborators, led by Tom Nichols, developed composite cognitive scores from the cognitive instruments that we administered during the HCA. He developed these cognitive composites in each of the memory and executive domains. Similarly, our collaborator, Carlos Cruchaga, contributed his expertise to calculate polygenic risk scores. He applied the standard practice of calculating polygenic risk scores with and without ApoE4 status in the data set. The goal of this analysis was to learn which, if any, of neurochemistry, cognitive performance, or genetics correlated with one another. The table shows that the strongest correlation of approximately 0.5 were between neurochemistry and cognitive function. These correlations are plotted in the 2 charts on the right and show that the more negative principal components for aging and AD are also associated with poorest executive function. Eva Rataj joined the Connectome team as the MRS expert at MGH. She published this paper, which suggests that the MRS-measured myo-inositol signal, or MI, is associated with glial activation. Glial activation was measured using 11-carbon PBR PET. 3T-MRS was used to measure myo-inositol in regions known to be involved in ALS pathophysiology, in the precentral gyri and brainstem, and in the precuneus as a control region. Whole brain voxel-wise correlation was used to identify brain regions in which there was an association between metabolites and tracer uptake. In the left and right precentral gyri, which are impacted by ALS, tracer uptake correlated with myo-inositol. This correlation was not statistically significant in the control region. In terms of ALS symptoms, upper motor neuron burden was correlated with increased tracer uptake and myo-inositol, and ALS functioning rating scale was correlated negatively with myo-inositol signal. This work demonstrates association between markers for neuroinflammation. For the AABC, MRS data, which were part of a separate project in the past, will be merged into the same project. MRS will be collected at the 2 sites that have 7 Tesla MR scanners, Minnesota and MGH. 150 participants will be scanned and have their spectroscopy scanned at each of these sites. This figure shows that the same very high spectral quality can be achieved at Minnesota and MGH, and in fact in Missouri, where I will continue to develop and use these protocols. We will continue to study the posterior cingulate cortex, which is illustrated in this parcellation. This image illustrates also that imaging outcomes from all modalities can be extracted for the MRS volumes of interest.